Let's talk about mitosis. So mitosis is the final step in a cell's life cycle if that cell is going to reproduce. And all the time before mitosis, the cell is in what we call interphase. Remember, interphase consists of G1, the first gap phase where the cell is growing up and doing all of its work, S, synthesis phase, where the DNA is being replicated in preparation for mitosis, G2, or the second gap phase, where the rest of the organelles and the cytoplasm multiply so that there's enough for the two resulting cells, and then finally mitosis. Now, mitosis consists of 4 phases. The cell moves from interphase into prophase. Prophase, the chromosomes are packed up, or this is my terminology, they're packed tightly so that they're ready to move. Before this, in interphase, the DNA is dispersed, diffuse. It just looks like an ordinary nucleus. So in prophase, the chromosomes are packed up, so we start to see that X shape. The nuclear envelope disintegrates because we're going to need all that space to separate the chromosomes and the organelles. We see these structures called centrioles, which are going to be used, or which are going to pull the chromosomes apart. And we see spindle fibers beginning to form and grow from the centrioles to the centromere, or the connecting point on the chromosomes. So what I've said here is nuclear membrane disintegrates, chromosomes appear, the X shapes that we're looking at, centrioles and spindle fibers appear, and chromosomes are packed and ready. So the P in packed and the P in prophase should help you remember what's happening here. So we're getting all prepared to do, oh, that would be another good P word, wouldn't it? Getting prepared to do mitosis. So the next phase is metaphase. And in metaphase, the spindle fibers, these green lines, attach to the centromere of the chromosomes, where the two copies are stapled together. And the spindle fibers also attach the centrioles to the membrane. So they're anchored to the membrane. And this is going to enable them to shorten the spindle fibers and pull the chromosomes apart as if with a rope. So spindle fibers attach to centrioles and centromeres. Chromosomes are lined up in the middle. So you will see a characteristic line of chromosomes in metaphase. And M in metaphase and M in middle should help you remember that phase. Now, in anaphase, the spindle fibers begin to shorten. The centrioles are beginning to shorten the spindle fibers, and the two parts of the chromosome are pulled apart. The two sister chromatids are pulled apart and move towards the centrioles at either end of the cell. So sister chromatids are pulled apart, A. Anaphase A should help you remember that. Finally, in telophase, which is the last step in mitosis, the cell begins to pinch into two, and this is called cytokinesis. Kinesis means movement. So the cell is actually moving and changing its shape now. The nuclear membrane begins to reform around the chromosomes, and the cell will pinch into two, and at that point those cells are now in interphase, and they're in their G1 phase. Okay, so we go from interphase of one cell, prophase, metaphase, anaphase, telophase, and telophase produces 2 cells which then start their life cycle in G1. I don't remember if I said, but the T in telophase and T for 2 should help you remember that one. Now let's take a look at the number of chromosomes that we have here. So there are 4 chromosomes. These are replicated chromosomes. We know they're replicated because they have that X structure. And so these 2 halves are identical to one another. So we have 1, 2, 3, 4 chromosomes. 1, 2, 3, 4 chromosomes. Here the sister chromatids are pulled apart. And once they are on their own, we no longer call them sister chromatids. They become chromosomes because they don't have a copy next to them. Once the cell finishes pinching in two, this is what it's going to look like, and it has 1, 2, 3, 4 chromosomes, and so does the other cell. 1, 2, 3, 4 chromosomes. The chromosomes will look like that, except that they will unpack during the G1 phase. Then during the S phase, they'll go through DNA replication. And finally, in G2 phase, the rest of the cell components will replicate, and then those cells will look like this and go through mitosis themselves.